We should get started. We are slightly late, but I hope we'll pick it up as this session progresses. So, well, hello, I'm Reganka Sir, and we are here for the first afternoon session on moving from bench to bedside in child mental health. And I'm pleased to introduce the first speaker. Dr. Catherine Pena is an assistant professor in the Princeton Neuroscience Institute and affiliate faculty at the Center for Health and Well-Being at Princeton University. And her talk title is up there. Welcome, Catherine.
Thank you so much. Thank you to Dr. Liwei Tsai and Dr. Ravi Raju for the invitation to speak today. It's truly an honor to be here. It's been such an incredible lineup so far of researchers and clinicians and public health advocates and doers. The whole day has been really inspirational. And I'm particularly excited to be in the session about translational work because while most of my work is preclinical using a mouse model, I think a lot about how to translate our work into the human realm and what this can mean for actually serving children who've experienced early life stress. So my lab uses a mouse model for understanding the long-lasting consequences of early life stress and adversity in the brain. And we're looking at this at a cellular level all the way down to the molecular level and epigenetic regulation of gene expression. So I'm going to rely a little bit on Ravi's introduction to epigenetics earlier and build on that. So thank you for such a beautiful introduction to epigenetics earlier. But first, we'll start with children. So early life adversity takes many forms. We've already heard about this throughout the day, so I'm actually going to skip over some of the background that's been presented before. But I will talk about some of the prevalence. So we think of early life adversity, we often think of abuse and neglect. This does affect about 1% to 2% of children in the United States. But this number is swamped by other forms of adversity that children experience. In particular, systematic adversity, such as poverty and racism, affects an estimated 20 to 40% of children around the country. Loss of a parent due to death or incarceration impacts another 7% of children. This is a huge stressor. As is loss of or displacement from one's home, which affects another 3% of children who've experienced homelessness at some point in their life in the United States. Unfortunately, another increasing trauma for children in the United States is exposure to gun violence, either directly or through active shooter drills. And a recent study just came out that maybe even these active shooter drills are traumatizing for children here, even though it can increase survival as well. Worldwide, IRC and UNICEF estimate around 30 million children have fled conflict and violence. Migrating children are at the highest risk for harm, abuse, deprivation, abduction, and other extreme stressors. And as we're well aware, this is a global problem right now, with the war in Ukraine going into its second year and other disasters around the world. So a better understanding of how early life adversity impacts brain development itself and increases some of the risks we've already heard about from physical health to mental health outcomes is really urgently needed. So we've heard about these ACEs or adverse childhood experiences before. The ACE that the impact of these adverse childhood experiences that I'm really focused on in the lab is understanding the increased risk for depression. So there's a 2 to 3-fold increased risk for depression among children who've experienced high levels of adversity. And you can see that even in the absence of recent stress, children who've experienced or individuals who've experienced high levels of childhood stress have increased rates of major depressive episodes. However, early life adversity also makes individuals more sensitive to stress across the lifespan. And there's this synergistic interaction between high levels of childhood stress experienced and recent stress to increase major depressive episodes and anxiety disorders. My group also saw evidence for stress sensitization in migrating children. So when I opened my lab in the Princeton Neuroscience Institute in 2019, I was very fortunate to have an incredibly talented pre-med Spanish major join the lab, Ana Elizabeth Sidamon-Aristoff, and she came into the lab and said, well, how do we, we're using mice, how do we stress a mouse, a pup? And I said, well, one of the main ways that we do this is through maternal separation. We take the pups away from the mom for a couple hours a day. And the next thing I know, she came back about 2 weeks later and said, I've been thinking a lot about what you said about maternal separation. And this is 2019, before the pandemic, right? So the news cycle at the time was really all about the US doing this at our border to migrating children, taking children away from their parents in immigration detention. So she came back and she said, I convinced the Spanish department at Princeton to give me a bunch of money to go down and interview families as they're released from immigration detention. Will you advise me on this project? It's one of these moments where you're like, wow, yes, I've never worked in human research before, but I'm going to say yes, and we'll figure it out. So we immediately called Dylan G at Yale, and she went down to the Texas-Mexico border 2 different times in summer of 2019. She interviewed parents of 84 children who were migrating from Mexico and Latin America. And one of the strongest things we saw was a correlation between the level of trauma these children experienced before migration and the PTSD symptom severity after they were released from detention. What we also saw was that children with at least moderate levels of trauma that they experienced before migration had the synergistic interaction that we expected with being separated. And so they had higher levels of PTSD symptom severity if they'd had at least moderate levels of pre-migration trauma. For better or worse, better for the children, worse for our statistics, we did not have children who experienced high levels of pre-migration trauma and separations. We don't know if that trend got even worse. But altogether, these findings really provide evidence against the policy of child separation, which is still happening to some extent in U.S. immigration detention. And it really supports humanitarian efforts worldwide focused on children and families displaced by disasters. Okay, but my lab is primarily focused on the brain mechanisms of this stress sensitization. And to do this, we turn to our mouse model. We have a 2-hit stress paradigm in mice. So our early life stress in mice consists of both maternal separation, as I mentioned. They're taken away from their mothers for 3 to 4 hours a day for about 10 days. And we also limit the home cage resources that the mothers have access to. So some fewer nesting materials are able to sort of like build these cozy homes for their pups. And then we put these mice also through adult stress, and in our lab we use a social stress for these adult mice to see if there's a sensitivity to that second hit of stress later in life. And what we find is that this mouse model mimics the clinical findings of increased sensitivity to future stress. So we actually don't see a lot of changes in behavior in the proportion of animals that show these sort of susceptible, depression-like, anxiety-like behaviors at baseline. But when we expose them to that stress later in life, they do show this increased sensitivity. One of the advantages of using mice is we can test sensitive periods for these effects. We cannot do this in human populations very easily at all. So we wanted to know whether it mattered when we stress these animals. Many animal researchers sort of assume that birth in mice is very similar to birth in humans and start stress paradigms shortly after birth. So we wanted to see, is stress right after birth in a 10-day period the most stressful experience to sensitize mice to later stress? Or maybe there's a period slightly later in development. And the reason we thought maybe this later period was important was because mice are born prematurely, their eyes are closed, and they really rely even more heavily than human infants on their mothers in this early period. And it's hypothesized that because they rely so heavily on the mom, it's actually a stress hypo-responsive period where they need to form these attachment bonds to the caregiver, to the mother, even if it's a stressful period. So it behooves them to actually not mount a stress response. And there's actually a lower cortisol, corticosterone stress response in early pups until about postnatal day 10. So we started our ELS2, our early life stress, in the later period at postnatal day 10 to see if maybe shifting this could actually change the sensitivity. And then again, put all animals through this adult stress. So we started looking at the males. This is sort of the graph that I showed you earlier. Standard root animals show some increase in susceptible behaviors. Interestingly, it was the animals that experienced stress in this later period after they have their eyes open. They're still pretty helpless. They're not even weaned yet. They're still very dependent on the mother for food and other aspects of growing up. But animals that experienced this exact same stressor, but right after birth, really didn't show this heightened sensitivity in the same way. Then we thought, well, females actually develop a little bit faster than males. Maybe female mice will show sensitivity to the earlier stress, ELS1. But we did not see that. And in fact, the female mice that we put through the earlier stress maybe even showed more resilience, but we're not totally sure of that yet. That needs to be replicated still. So in both males and females, it actually looks like there is a stress-sensitive period in development, but it's this later period of juvenile development. So when is this in humans? This matters a lot when we're thinking about these preclinical studies. And of course, all across brain development are important processes happening from fetal development all the way through adolescence. And there may be multiple sensitive windows depending on the processes the brain is undergoing. However, there is mounting evidence that the first week of a rodent's life is much more like the last trimester of human gestation. Still important, but different. And it seems like the stress-sensitive period that we have found in rodents actually maps onto early childhood of human development based on some of these brain processes happening. So this is our best guess that our stress-sensitive period really is early childhood development. Okay, so the next question is, where do we look in the brain for some of these effects of sensitization? And of course, stress impacts the whole brain, many systems all over. But the regions that my lab is really focused on are reward circuitry. These regions important for learning and motivation and reward. And we know a lot about how childhood adversity in humans does affect these regions. So poverty and childhood adversity sort of changes the course of development of these dopamine-rich regions in the ventral tegmental area. So the ventral tegmental area, this is a human brain and a mouse brain, is the seat of all the dopamine neurons in the brain. And one of its main projections is to the nucleus accumbens in mice and the ventral striatum in humans. So there's blunted development of these dopamine neurons in children who've experienced adversity. There's altered dopamine response both to stress and reward, increased stress response, decreased reward. And some of these changes predict anxiety and depressive symptoms. So we were really interested in looking at what's happening within these reward and dopamine circuitry. And we've looked at this at the level of cells. I'll take you on a journey deeper and deeper and smaller levels. Level of cells, and then within these cells, what's happening at a molecular level to genes that are being expressed, and then how these genes are being regulated by the epigenome, their physical three-dimensional architecture that the genes live in. Okay, so first, cellular microcircuits. We really wanted to know, are cells activated in response to early life stress, reactivated by adult stress, and is that some mechanism of this hypersensitivity to adult stress? And this actually turns out to be a challenge. So neuroscience is really good at measuring cellular activity in real time while something is happening, and very bad at knowing what cells were active previously. But we took advantage of some advances in transgenic mouse technology, if you will, that allows us to permanently tag cells that are responsive during a specific experience, a specific window in time. And we can tag them, track them, manipulate these cells, and isolate them to look within them later on. So I'll keep coming back to these mice. This is what it looks like. We can actually put a fluorescent tag that glows green on cells that are active during specific experiences early in life. So we're tagging early life stress cells or control population of cells in standard rid mice. And then we can use other markers of activity to look at how these how the brain is responsive to later stressors, so adult stress, and then how these cells that are responsive to early life stress might be reactivated by adult stress. Okay, so we use these mice to test two different hypotheses. The first hypothesis is that maybe prior experience of early life stress contributes to overall more cells being activated in a brain region. And we looked all over the brain. We did not find a lot of evidence for this hypothesis. So we tested another hypothesis, which is that maybe early life stress activated cells are preferentially reactivated by adult stress. And here we do see evidence, particularly within this brain region, the nucleus accumbens, which receives a lot of the dopaminergic projections in the brain. And you can see one example here of all the cells in the nucleus accumbens that were active during the experience of early life stress, and then a number of cells that are reactivated in response to that adult stress. And this was a significant interaction between these two experiences. And it was more in the nucleus accumbens and in many of the other brain regions that we looked at. So this is really exciting, but we wanted to know, is that actually relevant for their behavior? And so we asked whether we could quiet these cells, the activity of these cells, during a stress experience, and whether that would rescue this enhanced sensitivity to future stress. So to do this, we took advantage of our transgenic mouse again. We put a custom molecule for the aficionados in the audience. We used an inhibitory dread under the control of the ARC-activated cells. So we put this custom molecule that allows us to fine-tune the activity of these neurons In the mice, we did surgeries at postnatal day 7, which is quite tricky. We induced the tagging on the last day of early life stress or in response to a control experience in standard reared animals. We tested behavior of all the animals 3 times. First at baseline, then after all of the animals went through this social stress, we tested behavior again. In this first experience of social stress, they were allowed to experience that stress fully. We just gave them a vehicle. So they looked at behavior again. And then we put the animals through this social bullying stress a second time. But this time we gave them the ligand to interact with this virus that we had given them to quiet the activity. So we tuned down the activity of these early experience responsive cells while they were going through stress the second time and tested behavior again. And we found that, in fact, yes, quieting these cells can ameliorate stress hypersensitivity. So here is our behavior at baseline with more susceptible. I've sort of changed the axes on these, but I can talk about specific behaviors if anyone's interested. With more susceptible, lower, and more resilient, higher on the axis. So as before, early life stress by itself didn't really change the behavior of these animals. When they experienced this adult bullying stress, they did show this difference, where early life stress animals were more sensitive. However, even though they experienced stress a second time, just quieting the activity of those early stress-sensitive cells rescued the behavior significantly. And we did not see this rescue in standard reared animals that had sort of a random population of cells tagged, which indicates that while quieting this random population doesn't affect depression and anxiety, it's not just like the number of neurons that we turn off, that these specific early life stress sensitive cells are really important, and the reactivation of those cells are important for the stress hypersensitivity. We next wanted to know what's happening within these cells at the level of gene expression in particular. So in my postdoc earlier, I did bulk tissue RNA sequencing to look at the expression of all of the genes in the genome in way too many samples across way too many brain regions, and I'm just going to give you a really high bird's eye view of some of our findings here. So we saw a lot of genes that were altered after early life stress by itself in a number of these brain regions, including the dopamine region, the ventral tegmental area, and its projection target, the nucleus accumbens. So a lot of long-lasting changes in gene expression related to early life stress alone. And many of these genes had to do with nervous system development, indicating that perhaps early life stress was actually changing the course of brain development itself. Unsurprisingly, brains are also responsive to adult stress, so a lot of genes are altered in response to adult stress also. But what really caught my attention was the number of genes changed by the combination of early life stress and adult stress. And these were largely unique genes that were being turned on or off, indicating that early life stress was doing something to gene expression, but sort of holding it in place also. And that break on these changes was finally released by a second hit of stress. There were latent changes in gene expression. And the genes in this population were enriched for a response to stimulus. So again, they're hyper-responsive to what's going on in the environment. We also wanted to know, are these gene expression changes, so I'll come back to this. To me, this problem of these latent gene expression changes looks like a question of how genes are being regulated, which is why we get to the epigenome in just a minute. But first we want to know, are these changes in gene expression relevant for mood disorders, anxiety and depression? And one of the fun things that the pandemic allowed us to do when we were cut off from our bench work was to really dive into some bioinformatic comparisons. So we took these changes in gene expression that we'd previously looked at, and we got our hands on a data set of gene expression changes that were related to antidepressant treatment response or non-response in humans. So this is a sample of RNA sequencing from white blood cells collected by Dr. Gustavo Trecchi's lab at McGill. They're control patients and then patients with major depressive disorder. They sampled gene expression at baseline, and then all of the depressed patients were randomized into escitalopram or desvenlafaxine treatment. And then at a second time point, RNA sequencing was done again, along with assessment of how effective those treatments were. And this allowed us to look at those who had undergone treatment and responded positively to the antidepressants, and those who failed to respond to the antidepressants. And we specifically wanted to know whether changes in the brain after early life stress might actually predict treatment response or non-response. And this is partly because there's a growing body of literature showing that depressed individuals with a history of early life adversity and trauma actually have a different course of depression, earlier onset, more frequent episodes, and also respond differently to antidepressants, which maybe is not surprising if the brain chemistry is changed. So we wanted to see if we could use these bioinformatic data sets to actually examine this question. Okay, so I'll walk you through what this analysis actually looks like. We wanted to look across all of the gene expression changes in the brain, and one of the ways we did that was with a rank-rank hypergeometric overlap analysis. Essentially, how we read this is if there's heat in the quadrants along that I'm showing here, this indicates genes that are upregulated in both comparisons or downregulated in both comparisons. If there's color in the opposite quadrants, it means there's an opposite regulation. You can actually sort of flip the sign on that to look at similarity. And if there's no color here in the purple, that indicates there's really no similarity at all between these two comparisons. So the comparisons we wanted to look at first were antidepressant non-responders compared to responders, and the effect of 2 hits of stress versus only adult stress. So does the difference of having prior early life stress actually map on to treatment response or non-response? We looked at this separately in males and females. In females, we actually do find that a history of early life adversity significantly biases the expression patterns in the brain to look like expression patterns, these gene patterns, of antidepressant non-responders, so failure to respond to treatment. Interestingly, there was really not a strong prediction for males. So this non-response is biased towards females. So this is in the this human data set with the SSRI and the SNRI. We also looked at some mouse data sets, which had tested a tricyclic imipramine or novel antidepressant subanesthetic ketamine. And we see these similar responses. So looking at tricyclics, again, females look much more like non-responders to these tricyclics, and males even look like they might respond, even with a history of early life stress. And then to ketamine, again, it looks like a treatment failure among females is predicted if you have a history of childhood adversity, whereas there's really no strong prediction among males that had experienced early life adversity for the ketamine treatment. So together, these analyses indicate that early life stress alters the brain at a molecular level that renders it less responsive to a variety of antidepressant treatments across classes. And sex differences in these effects may help explain sex differences in depression and treatment efficacy, which really highlights the need for novel antidepressant treatments that are more efficacious in this high-risk population. Okay, so I promised epigenetics in the title of this talk, and I want to remind you of these latent gene expression changes. So these latent behavior changes and these latent gene expression changes. We want to know how these are controlled. And the level of genes is really controlled by the architecture of the genome. So now I want to convince you that the 3D structure of DNA matters. We normally think of DNA as just A, T, C, G, the sequence itself. We sort of think of it linearly, as I'm showing right here. But actually, they're wrapped around these balls of proteins called histones. And this helps keep them compacted and organized in the cell. And these histones have these little protruding tails that you can see here, and these tails can be decorated. These are post-translational histone modifications or chromatin modifications. And where these decorations are and the types of molecules they are really matters for the three-dimensional structure of DNA, either really keeping it compact or allowing it to be a little bit looser. And if it's really compact, I'm borrowing the library analogy that Ravi had earlier, this book is closed, you cannot read these genes. However, if it's decorated with other decorations, including this molecule I keep coming back to, histone 3, lysine 4, monomethylation, so single methyl group attached to the 4th lysine of the 3rd histone. This predicts sort of a more open conformation, and this is a book that's available to read. Okay, so we've talked a little bit about sensitive periods. Are there sensitive periods in the epigenome we might care about? And it turns out that there are a few studies looking at potential sensitive periods for stress to impact the epigenome in humans. And in particular, across different types of adversity, Aaron Dunn found that early childhood was a sensitive period for all these different types of adversity to increase changes in the epigenome. We also think that our mouse model is affecting, is approximately the same time as early childhood. So what about mice? What's happening in the epigenome in this period in mice? There's a recent study that I got really excited about. This is sort of simplifying it in many ways, but they looked at these distal regions, these sort of non-coding regions of the genome that don't have protein-coding genes in them, and looked at how open or closed these regions were across development. We see huge changes in the opening or closing in both directions of the epigenome between a mouse 1 week after birth to 3 weeks after birth. However, then it was pretty stable. And this is really interesting to me. So the largest change in chromatin, so chromatin is DNA plus these histones wrapped around, was between postnatal weeks 1 and 3, and then it's stable. And this is also our stress-sensitive period. So it might be that we really have hit a epigenetic stress-sensitive period in brain development here. Okay, so how do we look at whether early life stress actually changes the epigenome? These decorations can be everywhere across many different parts of the chromatin. And so we wanted to look not just at one at a time, but at all these decorations, all these histone modifications all at once. So we used mass spec. And I won't show you all the results, but one of the things that stood out was that in 2 different brain regions, the ventral tegmental area, the dopamine region, and its primary target, the nucleus accumbens, we see increased levels of this particular modification, histone 3 lysine 4 monomethylation. And this is the mark that sort of marks these open chromatin regions. We saw this by mass spec, we confirmed it by Western blotting, and we also see it looking across the genome with chip sequencing. This is particularly convincing to me because I was thinking initially of the opposite that we would find. So finally, all these things pointed that I was wrong, and I really needed to look at the data and listen to the data. And in fact, early life stress was opening up the epigenome. Okay, so why was I interested in this particular mark, H3K4 monomethylation? And I'll come back to this 3D architecture. So we have closed chromatin, our closed book that you cannot read. We also have open chromatin. You can see here, maybe this is a loop beginning to form, this open chromatin, where it is open, but we're actually not reading the genes when it's like this. And in order to actually get gene activity, we need to physically bring these distal regions of the genome, called enhancers, to the start of the gene, to the gene promoters. And then once we bring the enhancers to the promoters, it brings everything necessary for gene expression. And now we can actually read the book. So in this case, the book is open, but we're not really reading it yet. It's just available. And interestingly, we call these primed enhancers and active enhancers. These open and primed regions are marked by this particular modification that we found to be increased after stress, H3K4 monomethylation. We also see it here. We can distinguish between primed and active by other modifications. Okay, so I got really excited about this. We have good evidence that maybe the epigenome is being primed by early life stress, making it more reactive potentially to future stress. But I wanted to know, does it actually open chromatin? And I have a really talented graduate student in my lab, Rebecca Rashford, who decided to answer this question and look specifically within cells that are sensitive to the early life stress experience. So she's taking advantage of this mouse model that I already introduced. And she took out the cells that were responsive to stress and the ones that were not responsive to stress. We looked at how open chromatin was in both of these samples using attack sequencing. Okay, so here's a sort of data-heavy slide. I'll teach everyone how to read it for those of you who are less familiar. Along the x-axis is going to be how open or closed the epigenome is after early life stress. So more open to the right, more closed with early life stress to the left. And on the y-axis is how sure we are of that finding. And what Rebecca found is that within these cells that are responsive to early life stress, we have about twice as much opening of chromatin as we do of closing of chromatin. And even more exciting is that when we compare these to the cells that are not responsive to stress, we do not see these same effects at all. In fact, those cells all sort of look the same. So it's really these stress-sensitive cells that also show these changes in the epigenome architecture. So this supports our hypothesis that early life stress primes gene expression response to future stress. But then we wanted to know, again, does this really matter? Can we artificially prime the genome to make mice more reactive to future stress at a behavior level and a level of gene expression? So we made a vector to try to artificially open up, deposit this particular decoration, this histone modification, and open up the genome. then we put the animals through mild social stress, targeting this time the ventral tegmental area where we have all these dopamine neurons. And this work was all done by postdoc Julianne Balweck in the lab. We find that mild social stress, among control animals that do not experience this stress, they show basically control levels of behavior. So just having this artificial epigenome priming on board doesn't really change behavior by itself. But when we do expose them to this mild social stress, we find them more susceptible. So the epigenome is being primed to make these animals more sensitive to that stress later in life. Although it's a big jump to go from the epigenome to behaviors, we wanted to look at next the output of the epigenome, which is gene expression. And here I'm going to show you these volcano plots again, sort of spewing data at you. And once again, we see that if we artificially prime the epigenome from development and then expose animals to this mild social stress, this primarily induces gene expression changes. And this is in stark contrast to what we see without the priming modification, which is primarily closed. Okay, so I know I'm out of time, which is perfect because we're at conclusions. We've seen that early life stress sensitizes both humans and animals to future stress. Early life stress primes behavioral response to future stress through reactivation of cells, particularly within the nucleus accumbens, and that quieting this activity promotes resilience. We also see that gene expression changes in the female nucleus accumbens predict antidepressant treatment failure. And the early life stress primes the epigenome to be more open and reactive to future stress with the role for this particular chromatin modification. However, I do want to sort of focus our last point on a point of hope, which is that if we can increase sensitivity to stress through these modifications, we might also be able to take advantage of this increased sensitivity and maybe test whether there's also increased sensitivity to positive interventions and enrichment. With that, I'd like to thank my fantastic lab and all of our funding sources and your attention.